Fatty acid oxidation explained briefly. Fatty acid oxidation is the mitochondrial aerobic process of breaking down a fatty acid into acetyl-CoA units. These acetyl-CoA units then move into the citric acid cycle. Other products from fatty acid oxidation include NADH and FADH, which are utilized in the electron transport chain. You may hear fatty acid oxidation referred to as beta oxidation due to the oxidation of the beta carbon of the fatty acid. So step 1 is lipolysis, and this is basically just fat breakdown, and this must occur to give us the free fatty acids. Otherwise, the fats are stored in adipose tissue and aren't readily available for cells to be used as fuel. So then that brings us to step 2, which is transport and uptake. Due to the negative charge of the free fatty acids, they cannot cross over the cell membrane easily. In fact, they require specific transport proteins to help them cross over that cell membrane to get into the cytosol of cells. And that brings us to step 3, which is transfer into the mitochondrion. So right here, this is whenever the free fatty acid has crossed over the membrane and is in the cytosol currently. So CoA ligase reacts with the fatty acid that entered that cell, and this forms long chains of fatty acyl-CoA or short chains of fatty acyl-CoA. The short chains can diffuse pretty easily across the mitochondrial membrane. However, the long chains must utilize the carnitine shuttle. Fatty acids must be activated before oxidation, and they utilize ATP in the presence of CoASH and acyl-CoA synthetase. Finally, this brings us to step 4, which is the beta oxidation of fatty acids. Once the fatty acyl-CoA enters into the mitochondrial matrix, beta oxidation can occur. And this occurs by cleaving 2 carbons every cycle to form acetyl-CoA, water, and 5-ATP. This brings us into a little bit more of an in-depth molecular look of what's going on. So the fatty acid is dehydrogenated by acyl-CoA dehydrogenase, forming trans-delta 2 ennoyl-CoA. This enzyme actually uses FAD as an electron acceptor and is reduced to FADH2. That's where that's going on at. And then we have the molecule is then hydrated to produce an L3-hydroxyacyl-CoA. And then that molecule is oxygenated to form 3-keto-acyl-CoA. This enzyme uses NAD as an electron acceptor, and it is reduced to NADH. The last step is going to be the cleaving of the carbons, and this happens between the carbon 2 and carbon 3, carbon 2 being a beta carbon and carbon 3 being the gamma carbon. This is of course happening on the 3-keto-acyl-CoA. Then a thiolase enzyme initiates this reaction each time a new coenzyme A breaks the bond on carbon 3. And this entire process will continue until the acyl-CoA molecule has been completely reduced to acetyl-CoA molecules. Thank you for listening.